So my paper is structural basis and regulation of the reductive stress response. So reductive stress is the depletion of reactive oxygen species, or ROS. This state impedes cellular signaling and can lead to cancer, diabetes, and cardiomyopathy. Cells work to counter reductive stress and elevate ROS by degrading the mitochondria gatekeeper, FNIP1. So binding of the FNIP1 to the E3 ligase of CUL2 Fem1b is unknown. And in this paper, their hypothesis is that the CUL2 Fem1b complex uses zinc 2+ to selectively recruit reduced FNIP1 to reduce the reductive stress. So mitochondria is known in chapter 7 of The World from Dust as the second origin of life. And in the mitochondria, there is a high concentration of iron. This iron reacts and creates ROS. So another way that ROS is taught is from sunlight, so the UV light. And we learn in Biochem 1 about how dangerous ROS is to our body, but we never learn about how dangerous low levels of ROS is to our body as well. So this paper follows how important the mitochondria membrane is when it comes to regulating the ROS levels in a decrease of concentration. So they detected that zinc 2+ in the Fem1b FNIP1 complex is what drives it by using X-ray diffraction, X-ray fluorescence, and inductively coupled plasma spectroscopy. And in using X-ray crystallography, finally, they found that zinc 2+ selectively recruits the reduced, not the oxidized FNIP1, which is shown in the mechanism of regulation later. So in these images, Fem1b is in blue and FNIP1 is shown in orange. So the interaction of these two complexes are present in the first figure, showing the zinc 2 attracting into each other. And this part of the active site is known as the cysteine-histine claw. And that is because the cysteines and the histines are working, their charges are attracting the zinc, which is attracting the FNIP1 cysteine and histines. So when they mutated the FNIP1's cysteine-histine residues to test this theory, they found that the unmutated FNIP1 binds higher than all the other mutations, which proves that the zinc 2+ residues fit into the cis-his claw in order to attract the two together. So directing to the FNIP1 substrate. They also noticed the importance of the tyrosine and lysine residues, and both are adjacent to the zinc interface. So the FNIP1, lysine, and tyrosine in yellow, and the Fem1b right here in blue, these interact together in order to bind and create a tighter bond. In figure C, we saw that the mutations of lysine and tyrosine impaired the binding of the FNIP1. This was then known as the KY finger. So they found that the FNIP1 only binds to a small part of the Fem1b complex, but the structural binding was well conserved. And we can see that because the most conserved is in red right here, and it's right where the FNIP1 fits. And so this begs the question of what else is in this substrate. So they found that the heaps is also binded as well as the FNIP1, and a mutation in the FNIP1 in the R126 residue interacts with heaps and creates an additional hydrogen bond, which then creates syndromatic development delay. So this disease linked to the Fem1b showed a major discrepancy. In vitro, the mutation shown in figure D does not appear to change anything. but in vivo, it showed to gain a function without the impact of recognition in purified settings. So this discrepancy between in vivo and in vitro, specifically of the R126 mutant, led to the idea of inhibitor. So using affinity and mass spec, they isolated the Fem1b unit from the CLU2 and then finalized their findings with fluorescent polarization. And this established that the R126 is critical for recruiting an inhibitor of ROS signaling. This inhibitor is Bex proteins. And in figure A, they ran Fem1b trial in red, Fem1b with inhibitor Bex3 in yellow, and Fem1b mutant R126 in blue, and the mutant R126 with the inhibitor Bex3. And they found that the Bex3 proteins protects the FNIP1 from Fem1b, but not from Fem1b126. So this is shown because the purple, blue, and red, or the two mutant Fem1bs and the original Fem1b without inhibitor, all fall within the same category. And the Fem1b and the inhibitor are shifted down. And then in H, they tested this and found that both the R126 and the C186 prevents the recognition of Bex in this binding affinity in comparison of the Fem1b. And finally, by titration, they concluded that it effectively inhibits Fem1b, but not Fem1b R126. And so in this sense, the Fem1b R126 inhibits the inhibitor. So for regulation, since Bex3 only binds to half of the Fem1b unit, when the reductive stress is sensed, oxidized FNIP1 gets reduced and binds to the Fem1b unit through zinc. It then goes through its cycle and creates ROS and elevates these decreased levels. So two major assumptions, two major problems with this assay research. One was an assumption that Bex protein was an inhibitor. They had given no background on how exactly they knew that this Bex protein was determined, but they had used it as an inhibitor and claimed that it was. They also jumped into the disease portion with no mention in the abstract or the thesis. They kind of threw in the R126 mutation as well. There was no evidence until after they had ran the trials that it was really the R126. But before running their trials, they were asserting that it really was the R126 mutation in this. And although it ended being correct that the R126 mutation inhibits the inhibitor, there was no background on this information. Thank you.